The second video is actually, well, still a bit of an introduction, but more detail on eugenics. So now that you've known a bit and definition of eugenics and different types of eugenics, so let's have a look at this study. Why is it being used and for what purpose? So most of the time eugenics is basically more kind of like an approach to achieve the goal of evolutionary processes, to know what happened and to more or less try to evolutionize the processes. So now that we have GE in terms of modern eugenics, so of course it would affect an organism's genome directly, and this would more or less save time because you don't need to wait for a long time, even for centuries for some animals, for them to evolutionize. So scientists of modern eugenics would just simply remove undesirable traits to be replaced or added with desirable characteristics. Now that we have CRISPR, that would be much more easier. So the complete description of eugenics, which is basically based on the complete science of refining genetic structure of an organism to improve its inherent characteristics. Now this is basically kind of like why GE is important in eugenics, but okay, let's relate it to human, which means that let's see if it is possible to clone a human or not. So we know that there are many types of GE. We have natural selection, we have selective breeding, we have genetic manipulation. So from the three, we know that okay, it starts from nature's own GE, which is natural selection or survival of the fittest, which means that those that survive the most, they are the one that is the supreme. So using natural selection, evolution still occurs, but it's very slow. But of course nowadays GE could help speed it up. For selective breeding, this is done by altering the course of natural selection. So this is also referred to as unnatural selection by using GE to create your own desirable traits. And of course, the true or the recent types of GE would be genetic manipulation, okay, current means. So various methods could be used, and that would include CRISPR, which will be towards the end of this chapter. There are various methods to be used for genetic manipulation, either to introduce new genetic material or altering existing material. And the last one, okay, that we have over here, so I leave it to the last, okay, is actually true GE. This is what will will be debatable. True GE. When I say true genetic engineering, it means that you create a whole new gene or protein or organism from scratch. So far, this is still under research, but probably who knows, okay, it may be a reality sooner than later. Alright, so let's have a look at an example of evolution of E-skin, or, well, you know what E means, okay, so basically, okay, we know that skin is an important part and the largest part of a human body, and skin is, of course, important. So to grow skin, especially for those burn victims, will take some time. So now, okay, what they do is that they do grafting, that is by tissue engineering. Then from there, they basically develop other artificial or better skin, not just the skin, the same skin that we have, but improved material and so forth. So that means that your e-skin doesn't mean that it is similar or same as your own skin, but it is made of a better material, right? And let's have a look at one other example, which is the smart heart. So those who have heart disease will know that if you were to replace your heart, you will have to wait in line, you will have to wait for donor, and for someone to donate the heart, which means that they have to die, or they would basically die of an accident or whatever, and then only the person willing to get a new heart, okay, will get it. So that would take some time, and the complication is that it may be incompatible, so it could endanger the recipient's life. So they developed a smart heart. More or less, the smart heart, on the outer look, it looks similar to our normal heart, but inside, again, it is basically a mechanics, or basically it is made of kind of like machineries. So you can see that they still have a left chamber, okay, all the right chamber and whatnot, okay, but motor, okay, is being incorporated to replace the original equipment that we have, okay, in the heart itself. So apart from that, again, this is basically how, okay, forming the tissue cells mesh with biodegradable scaffold, which means that how you could create a biodegradable scaffold internally inside your body. So you can just have a look on this picture and even in your notes. I'm not going to go into details because this is actually a very complex process that they are doing nowadays. But the reason of having the biodegradable scaffold, okay, is that so that your internal tissue or internal organ, okay, could heal beautifully, but at the same time the scaffold would later on be degradable. Let's move our focus to cloning. So you know what cloning is. So let me just remind you or refresh your mind. So cloning is a process, okay, to create identical copies of an organism, and you could also create it just like that. So there is two ways cloning could be done, and of course that would bring us to two different definitions. But first, with cloning, we know that it might be possible to be carried out, and some extraordinary advances of GE could also be done. Okay? And this will, of course, in turn speed up research and further manipulation of desirable traits. So, this is what again I was talking about on differences in cloning. When you clone a gene and when you clone an organism, it means two different things. When you clone a gene, you just isolate an exact copy of a single gene from the entire genome, not including the whole genome, the whole organism. So the sequence from the DNA of the gene usually are the only one copied and inserted into the vector. And those are the ones that is being cloned, not the whole part of the genome. As compared to cloning an organism, whereby you make an exact genetic copy of the organism because you clone everything, the whole genome. Okay, so those are the differences. Remember that. Now, cloning an organism also refers to three different procedures and goals. So let's have a look. The first is embryo cloning. So of course it is a medical technique, and this is done to produce twins or triplets in the lab, and also those that desire twins or triplets not by nature. So duplicates of nature process is done by removing one or more than one cells from the embryo to be encouraged into developing duplicate embryo. So it results in cloning of basically a clone or twins with identical DNA. Second, which is the adult DNA cloning. So this is a method to produce duplicate of an existing organism. This is the method they use to produce dolly. And the third one is therapeutic. So as the name says, okay, this is, well, the method is almost identical to an adult DNA cloning, such as number 2, but it is transplanted back into the person for therapeutic purposes. So the aim is to produce a healthy copy of a sick person's tissue or organ, and especially used for gene therapy. So these are the three methods in diagrams. So probably you can see it better and see the differences in terms of how it is being done exactly. So probably what you could notice from over here again is that again human reproduction, the normal one again that we go through is as such. Human cloning or reproductive cloning means that basically okay, you need a somatic cell nucleus plus nucleated egg. Okay, so you remove the nucleus and you basically just fuse it together to produce a zygote, get the blastocyst and implant it into the surrogate mother, and of course give birth to the baby. Now for the therapeutic cloning, this is more or less where it is a bit different than these two. So the first part is similar to human cloning, but okay, the the next step okay is that once you get the blastocyst, okay, you produce embryonic stem cells. The harvest basically is being used to produce other tissues or other cells, and it is not being used to produce another human being. So more or less, okay, the second method or the first method that the doctors use to produce twins, if not by nature, is as such. So they make sure that fertilization occurs, and this is actually with the help in the lab even, such as when they create in vitro baby. So the same thing again for number 2, okay, which is the to create dolly. So if you remember the method that they use okay, is nuclear transfer. So you have an egg, you remove the egg nuclear material, okay, you have a donor cell, so this is enucleated egg. So you fuse them together, and then of course hopefully you get a zygote, and then you just transfer it to the surrogate mother. Now before we proceed further, so understood or try to understand those three processes, because second activity is actually involving or related to the three processes that I've talked about earlier on. So second activity is on the three reproduction processes. Do you understand them? So if you don't, or even if you do, try to do this activity. So hopefully it would enhance or help you to understand it better. Right. So find some information on first advanced Advantage or advantages of reproductive cloning versus human normal reproduction. Secondly, of course, once we have advantage, find some disadvantage or disadvantages of therapeutic cloning. So we know that therapeutic cloning has lots of advantages. What about the disadvantage or disadvantages? So jot down the information, only these two, and you can paste or upload it on e-learning under activity 2 reproduction process, which is directly under this video, the link. So you do that, and then later on proceed to video number 3.